We are live on YouTube. Going live in 5, 4, 3, 2. Okay, we're going to get started with this week's Miami Global Brain Tumor Symposium. Welcome, everybody, Wednesday afternoon or evening, depending on where you are. Just some quick introductions. My name is Michael Ivan. I'm one of the neurosurgeons here at the University of Miami, specializing in brain tumors and skull base, and director of our brain tumor research. I'm joined tonight by Dr. Komatar, who's a professor and program director. Also, his birthday tonight, so happy birthday, Dr. Komatar. He's the director of surgical neuro-oncology in the UMBTI. Dr. Morcos, also professor and co-chairman of our department and director of our cerebrovascular and skull-based program. And Dr. Benjamin, assistant professor, brain tumor and skull-based surgeon, director of our skull-based laboratory. This is our 33rd week putting together our symposiums, and I wouldn't be able to do it without that help from our administrators and the support from the University Sylvester Comprehensive Cancer Center and the Department of Neurosurgery. Thank you all, the administration. If anybody has questions or about tonight or future symposiums, please reach out to us. We're on social media, Twitter, YouTube, Instagram, Facebook, and also this is our website. You could find information about this symposium and others on that location. And just as, for instance, we have several symposiums going on this month, a pediatric symposium happening on Monday, talking about craniosynostosis with Dr. Proctor. And then Dr. Morcos is starting back his cerebrovascular and skull-based symposium, which will be on February 18th on the Thursday. So third Thursdays of the month, where he's going to be discussing mastery in cerebrovascular surgery with obviously some of the masters that you could see there. So please be sure to tune in. As far as the next brain tumor symposium, we'll be doing the first Wednesday of every month. This is a teaser for next month. We'll be welcoming Dr. Cheng from UCSF, just a pioneer in the field of neuro-oncology, and she'll be talking about advancing imaging in gliomas. So for some housekeeping for tonight's session, please use the Q&A button. We try to make this as interactive as possible. We don't offer CME, but you will get an email documenting participation, and please be sure to like, follow, and share us on Instagram, Twitter, and YouTube so we could share the great knowledge that we're going to be listening to tonight from our key speakers. Tonight, we welcome two panelists, Dr. Ene, who's joining us from Texas. He's a brain tumor specialist and a fellow at MD Anderson, doing his research year on viral therapies and oncolytic therapies and gliomas. He just finished his residency last year at the University of Washington and did some outstanding work there. And we're also joined by Dr. Shah, who's a chief resident here at the University of Miami, also has a strong interest in viral therapy, spent a year working with Dr. Kasahara on some oncolytic therapies while he was here, and now is going to be leaving us this year to go to the NIH to spend a year doing additional viral work in Washington. So they'll be presenting cases, and I thank them both for joining us tonight. Tonight's keynote speaker, Dr. Kioka, is a legend in neurosurgery, and we're very, very honored to have him join us tonight. As you can see, he's professor of neurosurgery at Harvard Medical School and the chairman of the department at the Brigham and Williams Hospital in Boston. He's a pioneer in biological treatments of the brain tumors, including the use of oncolytic viruses and gene and stem cell therapies to treat tumors. He's from Italy, but then moved here and initially started his education at the University of Texas, where he got his MD, PhD, before going to Boston and getting his residency at MGH. He stayed on as faculty before leaving to become the chairman at Ohio State, and then returned to Boston to be the chairman at Brigham and Williams in 2012, and has just done a phenomenal job since he's been there. His research interests are focused around gliomas, as you'll hear tonight, and he has multiple patents and papers, too many to count, on pioneering oncolytic therapies for gliomas that have really kind of set the stage for what the future generations will do. He's been continuously funded by the NIH since 1996, and is published in the highest impact journals such as Nature and Molecular Cell and PNAS. He also serves on multiple editorial boards such as JNS, Molecular Therapy, Neuro-Oncology. He's the past president of Society for Neuro-Oncology and on multiple advisory boards for both the NIH and brain tumor organizations. He was also elected into the National Academy of Medicine in 2014. Dr. Kioka, I just want to thank you so much for taking the time out to join us tonight and share with us your thoughts on treating gliomas. You're muted, but we could see your screen. Perfect. Oh, not yet. Still can't hear you.
Can you hear me now?
Yes.
Great. Listen, I am very excited to be a part of this symposium. I've heard a lot about it. I'm very happy that I'm finally participating in it. I'd like to thank Dr. Ivan, his team, the other co-directors, Dr. Komotar, Dr. Morkos, Dr. Benjamin, and it's a real pleasure to be here. What I'll be doing tonight is telling you how I apply my clinical enterprise, my clinical interests to my research interests. And one wise man once told me, if you could combine your research with what you're doing in the operating room, you'll have the best of both worlds. And that's what I'll tell you a little bit about today. So these are all my disclosures. I have many, as you can see here. So I clearly do not need to give this slide to the audience. You all know that with glioblastoma treatment, we try to do a gross total resection of a contrast enhancing area, or even a supramaximal resection, even remove some of the flare, and then patients undergo the stop regimen. As you know, Gliadel, Avastin, TTF fields have all been FDA approved. It can also be used. But all these tumors do recur within 6 to 9 months. And the median survival time from this initial diagnosis has been reported to be about 15 months. But this can vary based on genetic and demographic markers. Regardless, survival past 21, 22 months is really rare. But it can happen, and it's very anecdotal though. And clinical trials of targeted therapies and anti-angiogenic therapies have not been very successful. And everybody's been very excited about immunotherapy, but those, at least the initial ones, have now also not been very successful. One of the problems of why immunotherapies have not been successful has been this finding that the immune landscape of different cancers, including gliomas and especially other solid tumors, can be relatively cold. In other words, these tumors do not have a lot of infiltrating lymphocytes, do not have a lot of T cells that target tumor cells. And that is a combination of both the fact that we treat these patients with high-dose dexamethasone or temozolomide, which are both lymphopenic. And we also, the tumors themselves are fairly immunosuppressive. And the mechanism of immunosuppression by the glioblastoma have been elucidated by many, but they have to do with the secretion of various immunosuppressive factors. They have to do with the fact that these tumors are also full with cells that are immunosuppressive themselves, like myeloid-derived suppressor cells, as well as the fact that there may be also a sequestration of immune cells in areas of the body that basically do not allow the immune cells to get into the tumor. So what I will try, if you have to remember any slide, this is the one, which what I'll try to tell you is that one of the solutions for this immunological coldness is to actually inject into the tumor potent cytokines via gene therapy, or you can also inject oncolytic viruses. And that by doing this, you can transform this cold immune desert into one that's infiltrated with lots of CD4 plus and CD8 plus T cells that have been called into action by the expression of cytokines or other viral vectors within this cold immune desert. And this was really shown to be true with melanoma. This was a paper that came out in Cell, where they had melanoma tumors that were injected with an oncolytic herpes virus, and showing that these tumors fairly rapidly became infiltrated with lots of T cells. So what I will tell you about today is the use of gene therapy using adenoviral vectors to deliver potent immunogenes at the time of surgical resection of tumors. And these injectates of the adenoviral vectors into the tumor cavity can make potent immunostimulatory proteins, which then cause immunostimulation and infiltration of immune cells into the tumor. And we have several of these trials here at the Dana-Farber and Brigham. Some of these trials are multi-institutional. The one that we'll start off with is this series of trials that were first phase 1 and then phase 2 of using this approach called gene-mediated cytotoxic immunotherapy and combined with standard of care. And this is done for newly diagnosed glioblastoma. So there's very few trials that are performed when patients are first diagnosed, and this is one of them. And then I will go and tell you about some of the trials we're doing for patients with recurrent GBM after they've failed surgery, chemoradiation. So the mechanism of action of this particular method, which is called gene-mediated cytotoxic immunotherapy, is based on the fact that one can use vectors such as adenoviral vectors that have been totally devoid, they've been basically removed of their essential genes, and you're just using these as carriers to deliver an immunostimulant, as I said, into the tumor. So one of the prototypes was actually delivering the thymidine kinase cDNA. Thymidine kinase is actually expressed by a number of different mammalian cells, but the one that's made by herpes actually is a potent antigen, but it also endows the cells to chemosensitivity to nucleoside analogs like valacyclovir. So if you give the patient or the animal oral valacyclovir, the thymidine kinase will turn this into a phosphorylated molecule that lives inside the cell, the tumor cell, in the nucleoside pools. And then what happens if you apply some type of DNA damage like radiation, the DNA polymerases in the cell will try to repair this by using these nucleoside molecules, such as valacyclovir phosphate. These act as false nucleotides. They get intercalated And when they do that, they stop DNA polymerase from repairing DNA damage, so that you get DNA chain termination, and these cells will die. And in the process of dying, they start making immunogens that can stimulate the immune system even more. So this is the actual schema of these trials, which have gone to phase 1 and phase 2, and now we're in the active planning for a phase 3 randomized trial. So patients come to your emergency room or to your office, but usually it's been in the emergency room with a mass that seems on MRI scans to be a malignant glioma. These patients consent for surgery, but at the same time, we also consented them to the clinical trial. At the time of surgery, when we resect the tumor, the pathologist has to confirm that this is a high-grade glioma. If the pathologist cannot confirm that, then the patient drops off trial. We cannot do the injection. If there's a confirmation by frozen that this is a high-grade glioma, then we inject the cavity with these vectors. And then again, for 14 days, the patient takes this oral nucleoside analog of valacyclovir that gets phosphorylated by this thymidine kinase. And now you start applying the pressure of DNA damage by giving radiation at the same time. And as you give radiation, these cells try to fix the DNA using valacyclovir phosphate, but die in the process. And then you continue standard of care temozolomide. So this shows the cartoon again. You've resected these tumors. You injected this vector into surrounding brain that contains some tumor cells. The TK gets made in the tumor cells. Now you give valacyclovir. These phosphorylated valacyclovir molecules cause DNA chain termination and death. In this process, you release tumor antigens that get recognized, and you get this immunogenic cell death that stimulates immune cell stimulation. So there was a phase 1 of this trial back in the early 2000s. Then we went to a phase 2 from 2005 to about 2012. This is multi-institutional. And at the end of this, we had 48 patients that completed all this. So in a prospective fashion, when we designed this trial, we wanted to somehow get a sense of whether it was efficacious. It's very hard to know whether something is efficacious compared to just historical controls. And so we had a database that was matching or was accruing patients contemporaneously to this trial. And this was done at a 5th institution. There was not accruing patients directly to the GMCI, but was accruing them to this database. And so we had a prospectively collected cohort of patients that were getting accrued at the same time as this trial from 2005 to about 2012 or so. And at the end of the trial, we could then compare the survival, the efficacy of these 48 patients with 134 patients that seem to be matching fairly similarly to these 48. And so this was the actual results that were published in Neuro-Oncology in 2016. And so here we show the overall survival in the patient group that was so-called GMCI plus standard of care, and that was 17.1 months, compared to standard of care alone was 13.5 months. That seemed to be significant. Same was true for progression-free survival, 8.1 to 6.5 months. So these curves seem to separate, which was interesting. We wanted to go back to see what was the patient population that separated these curves. Was there one particular characteristic that separated these curves? And so we did an analysis of this. And when we went back and called out the patient population, it seemed that the one patients that had a gross total resection were the ones that really were causing this encouraging survival benefit. So here in solid line, I'm comparing the patients that had a so-called gross total resection. There were 18 patients in this group of GMCI plus standard of care. The median survival time was 25.1 months. In the standard of care alone group, the median survival time was 16.3 months. If you looked at the patient that had subtotal resection with nodular disease left behind, you completely lost survival benefit in the GMCI plus CSOC versus SOC group with the survival being 10.6 versus 12.8 months. So again, it seemed that in the patients that had GMCI, the ones that really benefited the most were the ones that are so-called grossal resection, with a median survival time of 25.1 months. That's higher than most, even more modern survival. So for example, in the TTF trial, the patients that had TTF, their median survival time was 22 months. And so this was even higher than that, if you wanted to make those comparisons. So I'm sorry. So questions you could ask me is, well, you've matched these patients. How did you match them? Did you match them for two things that we know are favorable prognostic factors? So one will be MGMT promoter methylation, and the other one is the IDH mutation, which is present in about 10% of glioblastomas or high-grade gliomas. So the problem was that this trial was run multi-institution between 2005 and 2012. So we did not really have IDH mutational status on any of these patients because the IDH mutation was only discovered in 2009, 2010. And so really clinically, it was not really used as a marker. And so we don't have that. And for only about a third of the patients, we did have an MGMT methylation status. And so here it is. And what you can see is that there was really no difference in the percentage of patients, whether that standard of care or the GMCI plus standard of care. There seemed to be no difference in their percent of patients in either group. So at least for a third of the patients in this trial, there seemed to be now a significant difference in methylation or methylation of the GMCT promoter. So we wanted, though, to still try to figure out whether there was any more of a bias and some other reasons why our survival curve seems so much better than the standard. And so recently, Annette Molinaro and her colleagues at UCSF have published a large database of patients, and they've looked at survival within their molecular subgroups. So here they're in this database to look at IDH, MGMT methylation. And so we've collaborated with Annette, and we wanted to compare, using these propensity survival matches, our survival curves to the ones in the database. So here what I'm showing you is that when we look at our database from the Brigham, our cluster control database, which only has standard of care, what we found is that this was very similar to the median survival from the UCSF standard of care group. So in other words, even with modern groups of patients that got treated in the area of the IDH or MGMT methylation, these two survival groups seem to be fairly evenly matched. When we looked at our GMCI phase 2 treatment group, median overall survival, and matched it to the database that clearly did not have GMCI, we still retained a significant difference. And again, this is our original graph from our paper. So even looking at databases that were collected in more modern times from other centers, we still seem to have in our GMCI group a difference in survivorship. So this is great. And as I said, we are now in the process of actually designing a protocol. It will be a phase 2 randomized trial using this GMCI for newly diagnosed glioblastoma compared to a randomization of patients that do not get the GMCI, similar to what I showed you before. And it'll be multi-institutional. We actually had a meeting with the FDA today for final designing of the trial. And hopefully that will get started sometime in the next year or so. In the meantime, though, clearly there's still patients that are failing this treatment. And so we wanted to know, why was their treatment failure? So we went back into the lab and asked in mouse models of gliomas whether there was any upregulation of immune checkpoint signaling. Now, we'll spare you the data from the mice, but the answer was yes. when you treated mice that had gliomas with GMCI, what you saw is that there was upregulation of PD-1 and PD-L1. And so based on this, and again, many other people are doing this now, we've decided a second phase 1 study where now we are not only giving GMCI, but also we're giving nivolumab as an immune checkpoint inhibitor. And this phase 1 study is being carried out through the Adult Brain Tumor Consortium, as well as under the auspices of CTEP and Bristol Myers Squibb. Advantagene is the sponsor of the GMCI treatment. And we also have funding for some of the science from this program project grant. And again, this is enrolling newly diagnosed malignant gliomas patients. They're getting the surgery plus GMCI, but then after chemoradiation, they get also nivolumab. And we are supposed to enroll 36 patients, and the 36th patient will be enrolled tomorrow, will be treated tomorrow. And so basically we'll have finished this trial. It took us about 2 years to get all these patients enrolled. And I have to thank Patrick Wen, who's the co-principal investigator in this trial with myself. And hopefully we will have data from this trial next year to share with you all. So in conclusion from this first part is that in a newly diagnosed GBM patient population, we show some encouraging survival in a multi-institutional phase 2 trial. We do see that GMC failures may be due to immune checkpoint signaling. So we are now completing a phase 1 of GMCI plus immune checkpoint blockade, as well as just doing a randomized phase 2 trial of GMCI alone, which hopefully will start sometime this year. And this took a long time to get going. Some of it had to do with funding, but the sponsor finally has gotten a nice infusion of cash from various sources so that we can now carry out this trial. So I'll stop here with the newly diagnosed GBM trials and go to the recurrent GBM population. As you know, this is a population that's more desperate. They fail surgery, they fail chemoradiation. Sometimes they fail other treatments. Sometimes they fail other trials. So what I will tell you about is a very similar approach where we're using an adenoviral vector to deliver a potent cytokine, and the cytokine is interleukin 12. Now, why do we want to deliver interleukin 12? Interleukin 12 was discovered in the late 1980s, early 1990s, and it was discovered, and it's been called the master regulator. The reason being is that it's a powerful activator of CD8 plus anti-tumor T cells and makes them make lots of interferon gamma. It also makes NK cells makes lots of interferon gamma. It makes B cells turn into plasma cells to make lots of antibodies. It turns CD4 plus cells into these Th1 CD4 plus cells that are also very immunostimulatory. It reprograms myeloid-derived suppressor cells, and it also has anti-angiogenic effects to interferon gamma. So really, a lot of the interleukin 12 activities are mediated by interferon gamma, which in by itself is thought to be a very powerful immunostimulator. So when IL-12 was first discovered, clinical trials in oncology started by delivering recombinant IL-12 systemically, but they were found to be way too toxic. Patients developed what's called a cytokine release syndrome, and therefore these trials were halted. Some have tried to deliver recombinant IL-12 into the tumor itself to try to minimize the systemic toxicity. However, the half-life was way too short and there was lack of persistence. So really the solution was to deliver IL-12 in a gene therapy modality by using vectors. And again, we go back to adenoviral vectors, again, where this vector can be engineered with a gene, in this case, the IL-12 gene. The vector is packaged, it gets down into the cell, injects the gene into the nucleus, and therefore you start making this gene, immunostimulatory gene like IL-12. So one of the other nice things about this particular modality is that there was still some concern that when you injected a vector with IL-12 into a tumor, that you would have IL-12 being made constantly. And so the second thing that was done was to place an inducible promoter upstream of this gene for IL-12. And this inducible promoter is called the Rio switch. And here it shows you some of the components. But basically, this promoter is off. Therefore, in its basal default system, the gene is not getting made. The only way the gene gets made or gets translated into protein is if you give an activator ligand. This is a small molecule that basically binds to some factors, coactivator proteins, turns on transcription from this promoter, and that turns expression of this interleukin gene on. So in mouse models of gliomas, what they were able to show is that when you compared this vector plus this activator ligand, it's called V stands for Veledimex, mice that had this vector that made interleukin 12 under control of this regular promoter and then had Veledimex lived more than mice that, for example, had anti-PD-1 alone, bevacizumab alone, or temozolomide alone. And this is sort of dose escalation of more doses of Veledimex in mice brains. So based on this, an IND was filed by this company called Zeopharm Oncology, and Zeopharm Oncology is a sponsor for the trial. And the trial, really, the initial trial was designed where we would go in and resect the tumor and then inject this vector. But the vector is off. The patient would have to take Veledimex by mouth. When Veledimex is taken by mouth, it should cross the blood-brain barrier into the tumor, turn on production of IL-12, and then you would get this cellular immune response against the tumor cells. So again, this was a multi-institutional phase 1 study. We participated from the Brigham, Cedars-Sinai participated, University of Chicago, Northwestern, and UCSF. Now this shows the study schema. Again, these were patients with recurrent GBMs. Some were first, second, or third recurrence. And 3 hours before resection, we gave them 1 dose of oral Veledimex. This is the oral ligand. The reason being is we wanted to really be able to measure this drug in tumor. So we wanted to ask the question, does the drug truly get into tumor across the blood-brain barrier? And so then 3 hours later, we resected these tumors and then free-handed injected this adenoviral vector into the tumor cavity. And then the patient took for 14 days this drug Veledimex that turns on transcription from this promoter, and then hopefully you make IL-12 into the tumor cavity. So this is a dose escalation trial. The first cohort got a dose of the drug called 20 milligrams plus the vector. This seemed to be relatively well tolerated, so we went to a cohort that 40 milligrams of the drug. This actually turned out to be too toxic. Patient did develop a cytokine-like release syndrome, and therefore we went down to 30 milligrams. This also was too toxic, and so the expansion cohort became 20 milligrams of the drug. And this paper was published, now it's 2 years ago, in the Science Translational Medicine. So the questions I asked was, number one, does the activator ligand truly cross the BBB? Does it get into tumor? And if it gets into tumor, does it actually turn on transcription of IL-12 after this was resected, injected into the GBM cavity? And so this is the sort of the key slide. Here what we show is that we're looking at patients that had 20 milligrams of the drug, 30 milligrams of the drug, or 40 milligrams of the drug. And as you give more drug, you get more peak plasma levels of the drug Veledimex. And at the tumor at the time of craniotomy, we can also measure drug into the tumor itself. And there's a dose, a response effect. The more drug you give, the more you find in the tumor. So yes, the drug gets across. About 40% of the peak plasma levels are found into tumor. The second question is, does the drug truly turn on transcription of this promoter and turn on expression of IL-12? Well, that can be measured in blood. There are ELISA assays. So baseline, here are the patients that have not had the drug. You do not measure IL-12 in their blood. Now you give them the drug, and about 12 hours later, you can see that you start measuring IL-12 levels in the blood. And the more IL-12 you give, the more drug you give, the more IL-12 levels go up. to levels that actually are fairly high at the 30 and 40 milligram doses, and that is probably one of the reasons why these patients got relatively toxic. These are fairly hefty doses of IL-12. You stop the drug though, these are reversible fairly promptly. The levels go back down, and so this is not a constant. This is not a constant effect. As soon as you stop the drug, basically these levels go back down. And we also asked the downstream effect of IL-12. I told you was interferon gamma. IL-12 makes T cells, NK cells make lots of interferon gamma, and so we also measure interferon gamma levels in blood, and the same thing happens. At baseline, there's no interferon gamma. However, when you give 20 milligrams of the drug, you do find lots of interferon gamma in the blood, and this goes up as you give more drug. You stop the drug, these levels come back down. So yes, this drug does get across the blood-brain barrier, gets into tumor, and then when it gets into the tumor, it does activate this promoter to make IL-12 interferon gamma. So the next question is, okay, this is a phase 1, but is there any evidence that there's survival efficacy? So these are our survival curves for all the patients that had at least 20 milligrams of the drug. So there were 15 patients in this group. Again, this is a group of patients with recurrent GBMs. And the median overall survival time was about 12.7 months, which was encouraging. But as you can see, there's a fairly wide range of 4.1 to 17.1 months. And one of the interesting things that we looked at was also whether there was any effect of dexamethasone. Dexamethasone is thought to be a fairly potent immunosuppressive molecule. And because of this wide range, you wanted to know what was contributing to this wide range. And what we saw that one of the biggest contributor was whether the patients had standard dose of dexamethasone, and therefore their survival was much less. And these are patients that had over 20 milligrams of dexamethasone throughout that 14-day time period where we gave them Veledimex. Or if for some reason the patients did not get that much dexamethasone, their survival seemed to be much better. And the median overall survival time in this group was about 17 months or so. So is there any post-injection tumor data? Do you have any data that in post-injection tumors there was actually a biologic effect? And so we did have some patients that after treatment you would follow. And as you see, typically you start seeing areas of enhancement occurring in the area of resection cavity. So this gentleman had a grossal resection. We injected the cavity with the vector. We gave him Veledimex, and then 4 weeks later, he starts having a nodule that appears actually in the parietal lobe, further up from where this injection was done. And then at week 8, there's an area here that's growing and starts to grow even more. Week 24, this area in the parietal, in the occipital parietal lobe still seems to be there. And so we took him back and resected this entire region, and then asked by doing multiplex immunofluorescence what type of cells were in these tumors. So here we have 3 different patients, and we looked at CD3 plus T cells. The CD3 plus T cell population, that's all CD8 plus positive, and then expression of PD1 in these cells. We looked at the Tregs by FOXP3. We looked at NK cells by CD56, as well as PD-L1 expression. And what you can see is that there clearly is an upregulation of more TILs, T cells that are infiltrating the tumor after injection than before injection. Now, this is several months later. These patients were resected about 4 to 6 months after injection. So there still is quite a bit of TIL infiltration within these tumors. But as you can see, these TILs are PD-1 positive, which may be an indication that they have become exhausted. At the same time, we also looked at tumor interferon gamma. You can measure tumor interferon gamma levels in these patients as an indication that somehow IL-12 had worked and that you had more interferon gamma still being expressed by perhaps the T cells within these tumors. And again, if you look at baseline versus post-treatment, there's actually quite a bit of interferon gamma still existing with these tumors 4 to 6 months later. So based on the fact that we do see increased PD-1 and increased PD-L1, similar to what I told you before with GMCI, we then designed a second clinical trial where we're now combining this with nivolumab in subjects with recurrent or progressive glioblastoma. I just submitted this paper, so hopefully it will get reviewed favorably. But basically, this is the trial schema that we did. So this was done in a neoadjuvant fashion based on the Tim Cloughesy data showing that if you gave neoadjuvant nivolumab, you get more of an immune response than if you gave it after in an adjuvant fashion after surgery. So similar to this, what we did is we gave nivolumab 7 days before the craniotomy. We still gave Veledimex so we can measure levels of the drug in tumor. We resect this tumor, we freehand inject the vector in the tumor cavity, and then we gave 14 days of Veledimex so that the patient could get interleukin-12 being made. And then we gave nivolumab to try to prevent that PD-1 upregulation that we saw. So this trial is finished, and again, we submitted the paper, and we'll see what the reviewers say. But just to give you a little teaser, one of the questions we asked is, do you actually show that you drop PD-1 levels and resect the sample? So a few patients still recurred after this. So we took them back to surgery, resected these tumors. So again, these are some of these immunofluorescence analysis. Here we're looking at PD-1 immunofluorescence. And each year is a patient where they only got 1 milligram of nivo, which is 1/3 of the dose. And there doesn't, maybe there's a little bit of an effect, but there doesn't seem much of an effect. We're looking at the white cells for PD-1 here in these tumors. But in this patient that got the full dose of 3 milligrams of nivo, this is how much PD-1 there was in this tumor. And these levels seem to have dropped quite a bit within the tumor itself. So in conclusion, what I've shown you is that you can use an adenoviral vector to deliver interleukin-12. You can do this in a regulatable fashion using a small activator ligand. When you do this, you can still see interesting biologic immunologic activity in recurrent GBM patients. There's some encouraging survival. Post-hoc analyses show that dexamethasone may inhibit these effects. And so we have done a phase 2 trial already, which just finished accruing, where dexamethasone was not allowed. And so those patients did not get Decadron. In this phase 1, we did see post-injection GBMs having inflammatory infiltrates, evasion due to immune checkpoint signaling. So we have done the combination phase 1 with nivolumab, which was presented as SNO, and the paper actually was finally submitted yesterday. So I wanted to give you enough time for questions, and so I was going to stop here. But I do want to thank a lot of individuals within our group that worked tirelessly in these approaches, and really Advantaging really is the sponsor for the GMCI trial. I've worked with this group for about almost 20 years now, and they've been fantastic collaborators. And Zeopharm is the sponsor for the IL-12 trial. So thank you for your attention, and I'd be happy to take questions.
Awesome. That was a great talk, and really just phenomenal work. I mean, you make it sound like it was fit into 40 minutes, but I know that's a decade of work at least that you just presented. So congratulations to your team on everything that you guys have accomplished. One question here we have from the audience was, as far as the timing of viral vector surgery-associated inflammation, does this affect the therapy and the efficacy of the therapy?
Yeah, that's a great question. And, that is something that we're struggling, because clearly with these vectors and with this immunostimulation, you get quite a bit of an influx of T cells and immune cells. And it varies depending on which treatment you use, but some of the more powerful cytokines do show this infiltration within a few weeks. And there you have to deal with the problem of pseudoprogression, because you do see an increased enhancement, just like the patient I showed you. And so there you're scratching your head. You talk to your colleagues in oncology. You say, well, gee, is this progression? Is it pseudoprogression? I've not really found a good way of knowing whether it's one or the other, unless the only way to really know is to take out the tumor and to see. But clearly we'll need to find better imaging or better modalities to try to discern the inflammatory response from true tumor progression? I do not have an answer to that. It may need one of your smarter people in the audience to figure that out.
One question I had was just, understanding everybody's innate immune kind of status and whether or not in any of those cohorts you looked at, did you see if it mattered on what the starting kind of immune system was in those patients? Now, obviously, it's how do you assess that? Is it neutrophil, lymphocyte? I mean, that's the most remedial way, but I think other ways to do that are available. And so would you ever think there's a day that you would then predict, okay, this person you need 30 of Nivo, but this person you only need 5 of Nivo, and to kind of get everybody, revved up to a certain level?
Yeah, I know. I think that's a great question too, and I think that clearly that also depends on whether you're newly diagnosed versus a recurrent GBM, and there's clearly a lot of evidence showing that as this disease progresses, you become more and more immunosuppressed. I think that even amongst recurrent patients that are relatively immunosuppressed, there are some that are more immunosuppressed than others. I think that some people have thought that interferon signaling, that maybe transcriptomics and looking at interferon signaling in the blood, the ability, in other words, to sustain interferon response, may be a way to assess the level of immunosuppression. And that's definitely true for the viral vector and gene therapy approaches. I think PD-1 is an interesting molecule because clearly it has failed in clinical trials for GBM, although we're still applying it together with an immunostimulant. The question with PD-1, I think, is still the timing. When do you give it? Do you give it in a neoadjuvant fashion? Do you give it in an adjuvant fashion after the fact? And again, the dosing, is there, there's a dose that's certainly been reported, but are there other ways of giving it? Will giving it intratumorally or in areas where there's antigen presentation be more effective for GBM than just giving it systemically?
And one last question from the audience was, with the new development of COVID-19 vaccines and the use of mRNA, do you think any of that research could be extrapolated for new viral therapies for GBM?
Yeah, no, definitely. And I think that we'll see many more. We may see in the future many applications where you take messenger and TAA, tumor-associated antigen messenger RNAs from tumors, and deliver those in nanoparticles into the circulation or in lymph nodes intradermally to get a reaction to the GBM rather than using peptides, for example, or vectors that deliver DNA. I think the company BioNTech, or the Pfizer one, and even Moderna, their original approach was not to go against coronavirus, was to make tumor-associated antigens using mRNA. And that's how they were able to pivot to coronavirus from that approach.
Okay, great. Shiva, you want to start first?
Yeah, sure. I'm going to share my screen.
Thanks. Go for it.
Hopefully you can see that.
Yeah.
Excellent.
Yeah, looks
good. So I'm going to thank you, Dr. Kyoko. That was absolutely fantastic. It's a morale booster for us that are interested in, working in oncolytic viruses to see that, there are some patients showing some dramatic responses. And so today I'm going to talk about some of the work that I'm doing in the lab. I work with Dr. Fred Lang, who's the chair here at MD Anderson, working on Delta 24 and looking at mechanisms that mediate susceptibility to Delta 24. I don't have any disclosures, but just a brief introduction about what Delta 24 is. It is a modified adenovirus. So wild-type adenoviruses are things that cause upper respiratory infections, conjunctivitis, pink eye. And the way these wild-type viruses work is they express a protein called E1A, which binds RB1. And when it binds RB1, it removes RB1 from repressing viral replication. So the wild-type virus replicates really well. But we've modified, and Dr. Fueyo and Dr. Lang modified, these adenoviruses, and it's Delta 24 because it's missing 24 base pairs on the E1A protein. And that's a mutant E1A. And what happens in normal cells is this mutant E1A can't remove RB1 from repressing viral replication. And so the virus cannot replicate in normal cells. But in cancer cells that don't have RB1, the virus is very promiscuous. It replicates the viral cell, the cancer cell dies essentially. And so using this Delta 24 virus, Dr. Lang and the team here at MD Anderson did a phase 1 clinical study on recurrent glioblastoma. And we had 2 arms in that study. The first arm did a stereotactic injection of Delta 24. There was a dose escalation compartment to this as well. And we just followed those patients long-term looking for toxicity and response. But there was also this window of opportunity arm where after we did a stereotactic injection of Delta 24 through an implanted catheter, 2 weeks later, we brought those patients back, did an unblock resection, injected some more Delta 24. And then we looked in the tumor to see if there's any biological effect. Is the virus still there? Is it replicating? Is there immune infiltration? And then we did the traditional studies like toxicity and survival. And what we found was that there are 2 groups of patients. There are patients whose tumors continue to grow, non-responders, and patients who their tumors essentially responded really, really well. And that really played out in survival. As you can see here, those patients who the tumors responded really did well. And we had 20% of patients with long-term survivors who lived over 3 years of recurrent GBM. And today, what I'm going to talk about, the case I'm going to present is this patient number 12, who had a really dramatic response to Delta 24. And so this is a 54-year-old female who presented from an outside hospital where she had a left parietal GBM resected. It was a subtotal resection. And this was followed by the standard stoop protocol. But then 2 months later, almost 2 months later, she came into MD Anderson with this lesion here with an MRI concerning for tumor progression, rather quick tumor progression. And obviously, there's always concern for radiation necrosis. And so we did a stereotactic biopsy that confirmed that this was an IDH wild-type GBM. And so she was enrolled in cohort A and taken to the OR for intratumoral injection of Delta 24. She got 10 to the 8 viral particles via an implanted stereotactic catheter. And she did great. The case went fine. She was discharged home after a couple of days. There were no issues with the case. And ultimately, she came back for follow-up a month later. And the follow-up MRI showed that there was increase in contrast enhancement, as you can see here. Quite concerning. It was a large increase in contrast enhancement just a month after we implanted Delta 24. Now, the thing is, she had no headaches, no nausea, no vomiting, no wound issues. Labs were within normal limits. But here you have a 53-year-old with an IDH wild-type background, and you're asking yourself, well, should we go in and take this out, or should we just observe? And so that's what I was going to ask Dr. Kioka. And you already talked about this briefly, because this is a problem for us in this field, is how do you make the decision to take the patient back to the OR for a resection? And, in case she had like a lot of swelling, would you use dexamethasone or would you use Avastin?
Yeah, that's a great question. And so what we've been doing as of late, because we've seen this a lot, and especially with the oncolytic viruses, because this is an oncolytic virus, it's not really a gene therapy, that's more of an oncolytic virus. So with the oncolytic virus, we see this a lot, and the higher the dose, the more you see it of the virus, and it usually occurs about 4 to 6 weeks after injection. And so initially we were taking all these out to study them. Now, especially if the patient has no symptoms, has no headache, no nausea, no vomiting, we've been looking at them. If they have any symptoms, we've been trying to avoid steroids, because when we've taken these out, we've seen in a lot of cases lots of T cells, lots of inflammation, and we know that if you give steroids, those T cells die off; they're not there. So we try not to give steroids, and instead we give low dose Bev. We've been giving like the 5 milligram per kilogram every 3 weeks cycle. And most patients just, if they have symptoms, those symptoms get better, dramatically better, and we just follow them like that.
Awesome, because that's what we did.
Yeah.
We observed her, and over time, you can see that there was a decrease in enhancement that kind of started around 4 months. So we waited about 4 months to start seeing a decrease that kind of stayed in place for over 17 months.
Was this without Bev? With Bev or without?
Without Bev. Without
Yeah.
Bev.
Nice.
Yeah.
No steroids?
No steroids. Very little edema associated with this. And so we kind of held off on that. So, she had a progression-free survival of about 3.5 years and almost 5 years before she, passed away, unfortunately, from recurrent GBM. But this was to us sort of an example of where, you have to make the decision in a case like this as to whether to go to the OR or whether to just watch. And in her case, just watching this pseudoprogression play out, panned out really well. And, holding off on steroids also panned out really well for her. But the other question that came up was, what about what's actually going on under within this tumor? And that's where this window of opportunity arm came in. When we take these tumors out on block, you can actually see a catheter here. And what we found was that within 2 weeks, there's still viral proteins that are present, but not really present at about a month. But what happens over several months in the patients who had clinically relevant responses, one of them we took back to the OR just to make sure it was actually progression, there was a significant infiltration of T cells and macrophages, as you can see here, suggesting that it's possible that the long-term survival is actually mediated by the immune response, not necessarily the actual effect of the virus directly on the tumor cells. But that's really hard to prove, right? It's very difficult to prove whether the extent of anti-tumor immune response can distinguish long-term survivors from short-term survivors. And part of the reason for that is some people say it's unethical to take these patients back over and over again. And logistically, it's difficult to do it. Financially, technically, very difficult to actually biopsy the microenvironment, especially if the patient's doing well. So what we did was we took this into the lab. We performed a window of opportunity concept in a preclinical setting to see if we can find a more accessible systemic biomarker that could tell us what's actually happening. Is there a response cytokine that's released? Are T cells going up? What is it actually about these responders that's actually causing this effect? And in this case, in mice, we can get tumors, we can get spinal fluid, we can get blood, so we can hopefully get an insight into what's causing a response or what's causing a resistance. And so to do this, we used a bilateral tumor model that I helped develop when I was in Eric Holland's lab at the Fred Hutch in Seattle. This is a model where you put 2 tumors in the animal's brain, one in each hemisphere, and genetically you can engineer just one tumor to express luciferase. And what happens is, after you treat one with radiation, you just follow the luciferase, and that tells you what the animal's immune system is actually doing to a tumor that never saw the treatment that you gave, either it's radiation or the virus. And that tells you how strongly the immune system is reacting to a tumor that never got treated. And so you see this MRI with 2 tumors here, as well as luciferase coming from just one part of the brain. And so in the short time that I have, I can't show all the data that we have, but I can summarize it for you. And what we're seeing is that when you treat one of these tumors with Delta 24, the oncolytic virus, it's really effective. It directly causes oncolysis, the tumor goes down. But it is this tumor that we never treated that makes the difference in survival. So some animals show that this tumor continues to grow. We call them the non-responders. And then in some animals, the luciferase signal drops significantly. And that really pans out for the survival because the responders do significantly better. But what's more critical about this distinction is now we have a window of opportunity to start distinguishing these 2 populations. You can now biopsy tumors, you can take tumors out, you draw their blood, you get CSF, because now that you can tell the difference between this animal and something that could serve as a biomarker for who's responding, and importantly, maybe predict all of this stuff before it actually starts. So I can't show you what we have in the blood and CSF yet because it's still really preliminary. But what I can show you is that in the tumors of responders, there's a significant pseudoprogression that happens in these animals. So there's a lot of macrophages infiltrating into these tumors. And these animals were both treated with Delta 24. And so what this indicated to us was that not everyone treated with Delta 24 can mount an anti-tumor immune response. And we suspect that the patients who can mount an anti-tumor response are the ones who would actually live long. And so in conclusion, what I would say from that study is that I think as your presentation pointed out, Dr. Kioka, neurosurgeons are uniquely positioned to provide insights into why these immunotherapeutics fail and why some of them work. And it's because we have our hand in the field. We have access to the tissue. We can treat these patients and then go into the OR and take those tumors out to see what's actually happening. And that involves including a window of opportunity, concept into the clinical trials that we design. But even more importantly, we can take this concept back into the lab to help us discover systemic biomarkers that could actually predict who's going to respond and who's not going to respond, going back to Dr. Ivan's question. And this will help us determine whether a patient will be a long-term survivor or not a long-term survivor. But also those biomarkers, if you know what they are and they're really potent, like IL-12, you can actually pick patients who are deficient for that biomarker and actually arm Delta 24 with those cytokines to see if you can actually induce an anti-tumor response. And so this is where things are headed for us in the lab, is to figure out what the systemic biomarkers are. And there are a lot of people working on this. And I just want to thank them very briefly before I stop. Dr. Fred Lang, who's our chair here at MD Anderson, is my mentor right now, working on Delta 24 people in the lab. the Brain and Spine Center here, as well as the department at MD Anderson. And these are my funding agencies, including an R25 when I was a resident, as well as the SPORE grants that is funding a lot of the studies here at MD Anderson. So thank you.
Congratulations. That's a very powerful model.
Thank you.
So you'll get some really good results.
Thank you.
Yeah, great job, Shiva. I mean, that was great work, and I can already think of another 10 questions you could answer with the model. So
Absolutely.
I'm sure you have your work cut out for you. So
Oh, absolutely.
great job. Ashish?
Sure. Before I begin, Dr. Kioka, did you were using your gene therapy model was a replicating viral vector, or was
Yes,
it
the ones I showed you were gene therapy, so they're not replicating adenoviruses. I did not have enough time to show you our oncolytic virus, the herpes virus trial that we were pursuing now, the phase 1. But those results are still maturing, so there really was not a lot to show.
Absolutely. I guess the reason why I asked that is because I was kind of going to go over a little bit of our Miami virotherapy experience and kind of get your opinion on what your thoughts are.
Sure.
And just kind of that was a great talk, Chiba, by the way. I really enjoyed that, and Dr. Lang's work is very impressive. So I'd love to get your input as well. As you guys know, virotherapy is kind of a big trend in GBM research. There's a variety of trials, different institutions with varying outcomes. Here's a recent publication we had in the neurosurgical focus, demonstrating the variable outcomes between different types of viral vectors for recurrent or newly diagnosed GBM. At Miami, we had the fortune of using the TOCA 511 replicating retroviral vector as a kind of prodrug activating gene therapy. So it's a lot of words, so I'll kind of hopefully break it down. Essentially, it's just delivering a transgene, which is the cytosine deaminase transgene, which then gets enzymatically converted to active chemotherapeutic 5-fluorouracil once you give the prodrug, a non-toxic prodrug like 5-fluorocytosine. So in essence, 5-fluorocytosine is an antifungal drug that's used over the counter. It's it's FDA-approved drug that crosses the blood-brain barrier. Once the cancer cells are delivered this kind of replicating retroviral vector, cytosine deaminase converts 5-fluorocytosine to 5-fluorouracil, delivering kind of an intracellular chemotherapeutic. So it's almost like a tumor-selective gene therapy. And the benefit of the replicating retroviral vector is it selects for the tumor itself because it only preferentially replicates in cells that are dividing, and the tumors also have kind of impaired antiviral defense. So this is kind of been proven in preclinical models. And their initial phase 1 data had some pretty decent results. Here is convection enhanced delivery of giving this replicating retroviral vector for recurrent GBM, and you see kind of the stark kind of radiographic response after giving one cycle of prodrug. And this is another patient who had kind of multifocal disease, and you can see like the lesions are having nice complete responses. And the trial was designed to kind of have a kind of resection on recurrence. So you could see, whenever they had, recurrence, you could see lymphocyte infiltration as well, and staining for the transgene in the tumor. So with these promising, promising preclinical and clinical data, this was kind of moved to initial phase 1 trials, which I'm sure you're aware about, that demonstrated improved survival in the recurrent glioma patients compared to matched controls. But recently the phase 3 data just came out. Maybe I want to say about a year ago, was presented at SNO, and then just got published in JAMA Oncology. But really, there was really no difference in overall survival between the control group and the treatment group and the randomized phase 3 trial. So this was like a huge disappointment, for us in the kind of gene therapy world, with demonstrating really no difference in overall survival. But of course, we were kind of blessed with the subgroup analysis demonstrating that there was a subgroup of patients with kind of IDH mutant gliomas that did benefit and had a stark improvement in overall survival for recurrent GBM. So that kind of brings me to my point, and this is kind of one of the case examples I wanted to show you to kind of get your opinion, because this is kind of bugging us on this kind of gene therapy, viral mediated gene therapy world. This is a patient who was our first patient treated with TOCA 511. Is a 70-year-old male who underwent surgery and chemoradiation in April of 2015 for this kind of frontotemporal GBM. Had a gross orbital resection and a good response, but as expected, had a recurrence, distant recurrence here with crossing of the corpus callosum involving the splenium. This patient was taken back for a repeat resection of this tumor, and then kind of the resect and inject paradigm. We were fortunate at the time to take different areas of the tumor, so we sampled different areas and kind of cultured them using kind of our patient-derived stem cell cultures. Those kind of derived from the Toronto group, Peter Dirk's lab. And we established that these were in fact kind of stem cells. They all were able to derive tumors in mice models. But you can see that each area of the tumor has a differential, survival when you inject them in tumors. So, the center of the tumor, was progressed more rapid than, the outer parts of the tumor and the enhanced edge. And then whenever we treated these patient-derived tumor cells in vitro culture with our replicating retroviral vector, the response rates were different between each different area. And this kind of brings me to the idea that, well, how do we account for intratumoral heterogeneity when you're treating patients with gene therapy? Certain areas of the tumor will probably be more susceptible to taking the vector, and some of them will have different responses. And I've seen that at least in clinical. You see it in the clinical data, and then you see it in the preclinical data. So, is there a way to have a biomarker for predicting which patients are actually going to benefit from viral mediated gene therapy? Are there certain restriction enzymes or restriction factors that prevent the virus from going into certain cells? This kind of hints at the kind of cold and hot tumors you mentioned earlier, but I suspect that this is kind of where the field is moving.
Yeah, I know, that's very interesting, and clearly, the problem of heterogeneity is going to be a problem that's going to, vex almost any treatment that we think of. And whether different tumors have different levels of immunogenicity, regardless of whatever virus there is. I wish I had answers for you. I'm not sure I do, but I think one of the problems, and your data is interesting, right? Because you just see that the center of the tumor is more responsive.
Right.
Did you look at things like proliferation rate? Do the tumor cells from the center have a higher proliferation rate than the ones from the edge? Because at least with the retroviral vectors, retroviruses need a proliferating cell to integrate and to do their damage.
Yeah, they all had a very similar proliferation rate.
Okay, it's not something like that. And I guess the other question I have, was there any difference in the receptor, the retroviral receptor, that in any of those cell lines?
Yeah, that was something that we wanted to check as well. There's a few the PIT1 receptor is involved with the receptors for retroviral vectors, and also we want to check to see whether or not there was some retroviral silencing machinery that may be also like contributing to preventing the retrovirus from integrating into the genome.
Yeah, so, you can do like vector copy numbers in the cells to see if there's any difference in integration rates.
And we've seen in different cell lines, different mice glioma, like GL26. I know you presented the data for your gene therapy model in GL26, but for example, GL26 is very, very resistant to our replicating retroviral vector, but but TU2449, which is kind of a VSARC murine glioma line, is much more susceptible to replicating retroviral vectors. So, there must be something, at the cellular level, which restricts these viruses from working. And I'm just curious, how do we?
Well, yeah, and the other issue too is that you take these cells from this environment, but in those environments, there's not just tumor cells. There's also mononuclear cells, especially myeloid-derived suppressor cells. Some people have said even 40 to 50% of the tumor is actually stromal cells. And those never culture out because you're trying to culture out the tumor cells, but those cells don't culture out. And so that's also something that's not really well recapitulated in some of the experiments. So I'm wondering whether organoids may be better. Could you make organoids from these tumors in these areas, from these areas?
Absolutely. Yeah. Sorry for the puzzling questions.
We're working on the organoid model. We'll get back to you on that.
Yeah. You do center organoids, enhanced edge organoids, outer edge organoids.
That's right.
Yeah, exactly.
That may be an even interesting model. Well, that's great. Great work.
Thank you so much. Thanks, Dr. Ivan, for helping mentor me through some of these difficult times.
Yeah.
Okay, great. Well, that was great information. Thank you again, Dr. Kyoka. I think if we don't have any other questions, I think we got to the questions here that on the side while you were talking. But Shiva, Ashish, thanks for your cases. And it's always good to know that we have a younger generation working on such complex problems like this, and to kind of continue on Dr. Kyoka's great work and some more clinical trials going forward.
I'm expecting you all to figure out things that we cannot figure out. Thank you.
Okay, everybody stay safe, and thanks again.
Stay safe. Thank you.
Thank you.
Bye.